it turns out that you can modify the side chain of lysine. And there are two different ways to do that: acetylation and methylation. In acetylation, you basically neutralize the charge by placing an acetyl group on the amino group of lysine. You have an amino acid side chain with no charge. So you've taken away that positive charge. Think about what that might do to the structure of a nucleosome. You can also methylate, and you can do monomethylation, dimethylation, even trimethylation, which fixes that charge in some way. So again, it changes the character of the side chain and therefore changes the interaction with DNA. With serine, it's different. You can introduce a negative charge. Serine is normally neutral. It's an OH. You can create phosphoserine that has a negative charge. And how is that going to interact with DNA? It's going to repel the DNA. So these reactions can either stabilize or destabilize the interaction of histone molecules with the DNA. It can even change the interaction of histones with each other. And if we look at places where histones are modified within nucleosomes, you can see with the blue where it's acetylated, with the red where it's methylated, and with the green where it's phosphorylated. So this is a particular set of histones at a particular place in the DNA. And these different modifications cause changes in the structure of the histones that alter how the DNA that is packaged in this region gets expressed. And so these marks, these modifications of amino acid side chains that occur in the histones, are a way of altering DNA expression without changing the DNA sequence. The writer enzymes are enzymes that mark the histones, that acetylate, that methylate, that phosphorylate, and those are only a few of the many different types of modifications that can be introduced. Eraser enzymes remove those marks. They take away the modification and return it to its former state. And finally, there are reader proteins that recognize the marks that say, okay, we need to shut down more the expression of this region of DNA or open it more. And sometimes reader and writer enzymes interact to create another layer of complexity in how DNA is packaged and how it is unpackaged, unwound for expression, or tightly packaged to prevent expression. And it turns out that these modifications are increasingly important, or at least we're discovering increasingly that they are important.